sense of what a privilege it is to work with Bob Lanza. Just extraordinary scientist. I'm just going to switch, hopefully, to my talk. Get a little bit of luck here, please. Goodbye to your talk file. And
any purple label that you changed.
Sorry. I tried to put a macro in. So as we get started here, let me, you have to go right back through these. I apologize. The perils of two talks. Thank you for your patience. I have to say that working with Bob has been inspirational, and that didn't work, did it? One more time. I thought I had it selected to slide forward. Translating science like this from the bench to the bedside is a challenge and a privilege. And I've had the opportunity over the course of my career at UCLA and at the Jules Stein Eye Institute to participate in some moderately impactful translational experiences, one of which was the anti-VEGF story, whereby we took some drugs together with Hal Barron and people at Genentech and translated the anti-VEGF oncology program, which is Avastin, into the anti-VEGF ophthalmology program, which is Lucentis. And we treated the first patients here at UCLA, first patients in the world here at UCLA in 1999 with that drug. And that was the first time we ever saw vision go up in patients with macular degeneration rather than going down. So we blocked the abnormal growth of blood vessels. And we took home a lot of translational lessons from that long and arduous experience of moving from really the basic science, vision science, all the way through to approval and now what is, I think, something like a $6 billion a year global drug, which unfortunately I don't own, nor does UCLA. I wanted to share some of those lessons with you or learnings with you. So Bob just very eloquently showed us that the advantages of human embryonic stem cell derived retinal pigment epithelium. Unlimited number of healthy young cells, potentially reduced immunogenicity. We can modify or modulate the in vitro, the differentiation, to control cellular and molecular characteristics and thereby optimize safety, identity, purity, potency, controlling pigment expression, perhaps gene expression. So the translational ideals may seem obvious, but for the students in the audience or those of you who are hoping to become translational people, these are the principles that have really, the learnings that I've really taken home. And the first one is get it right. You absolutely need to get it right the first time here. You cannot, don't have the wrong hypothesis. It means choose your collaborators carefully and choose your targets well. Your target condition should be very, very carefully chosen. It seems obvious that improved basic science understanding of disease allows for the development of rational treatment strategies that are potentially safer or more effective or less risky. But there's a lot of detail here, and the flyby is sometimes missed, this global perspective of choosing carefully what you're going to study. Similarly with a therapeutic agent, particularly with this one, with a highly complex biological therapeutic, probably the most complex ever tried in humans, the formulation, the dosing, and the delivery details need to be manipulated and studied to optimize success. Just to share with you, we took all these great stem cells that Bob created for us, these RPE that are just beautiful, and we took our usual subretinal injection kit, and we just passed through the injection kit, and they all died. So the biocompatibility was really important to do, and we worked together, great collaboration with Bob and his team and our team at UCLA, and we were able to optimize this delivery system to make it work. And partly through how he manipulated it, partly through how we manipulated it, the collaboration was really key there. Now, I'm going to talk to you about some of the strategic obstacles, and I apologize if my slides are all different. I threw together bits and pieces of different talks to sort of give you an overview today. There are some big strategic obstacles for translational research, moving something from the bench to the bedside. Number one, first and foremost, is regulatory. And I have found that the FDA is actually pretty good to work with. They're reasonable, they're smart, they respond, and if you are simple, clear, and well substantiated, and you make a good case for your study for safety and tolerability, you can get into the clinic. And we, working together with ACT, were able to guide this IND, shepherd this IND through by simplifying and being clear. We're treating very end-stage eyes, so we're limiting the downside in terms of functional loss, vision loss. We have very pure cells, as Bob showed you, so the tumor studies, preclinical work was really strong. And we're immunosuppressing our patients, so we're mitigating against rejection. Those are all really important pieces as you go forward. There are lots of controversies that I won't go into in detail around whether RPE should be injected in solution or in sheets or in scaffoldings, whether Brooks membrane can tolerate it, and I'll get into some of that with you. The bottom line is you can't study multiple variables for the first study. You have to study one variable at a time, and that was the trick to getting into the clinic with the FDA. Tactically, it's incredibly important to have departmental and university support. The Jules Stein Eye Institute is an incredibly special place. We have an unbelievable vision science program there. And we have a really good clinical research program now. Multiple clinical trials, some of them have been described to you already, multiple basic science laboratories, and so there's an appreciation for that. But more importantly, we have great leadership that supports innovation within the Eye Institute. So it's a very special place. For those of you who are interested in translational science, we have our own clinical research center, and it's really a special place. And I want to thank the coordinators that are here today from the CRC in the Doris Stein building that have really made things happen. Now, for those of you who don't realize it, UCLA is a pretty special place too. UCLA came together, the UC came together behind this trial in a really special way. They did the right thing. We were able to assemble all of the relevant groups, whether it was the IRB, Contracts and Grants, University Office of the President, everyone, the escrow group, everyone came together in a very special way to make this happen very quickly and very collaboratively. And it speaks to our privilege of being here and being able to do that. And so to keep going with the obstacles, Bob mentioned immunogenicity, rejection, tumor genicity with teratoma, decreased visual function. These are all things we'll come to. Survival after rejection, compromised support system. The eye's like a camera. It's also like a room. The wallpaper that lines the inside of the room is the retina. Just deep to the retina, to the wallpaper, is the retinal pigment epithelium. The retinal pigment epithelium is absolutely mission critical for vision and the health of the retina. And the retinal pigment epithelium creates and lives on this layer called Brooks membrane. And Brooks membrane is an acellular basement membrane and elastin and other things in there that needs to be there for cells to attach and survive. And there's a lot of controversy around that we'll come to. Integration into native RPE layer and attachment to Brooks membrane, rescue of overlying photoreceptor compromise, in other words, making the photoreceptors healthier, and improve function. These are all potential biological signals. These are not things that are primary objectives of the first study. These are not primary outcomes. These are maybe we'll see them, maybe we won't. And if you read our paper in the Lancet, we saw some of these, but we don't know what quite to make of them yet. But really it's safety and tolerability. So why the eye? Why is the eye low-hanging fruit? Well, it's an ideal target for complex biological therapeutic agents. You know, gene therapy is here today in the clinic, in the eye, and working. There are patients who have Leber's congenital amaurosis who have been treated with AAV and an RPE65, which is one of the enzymes in the visual cycle, that are 4 years out with improved vision. 4 years out. Just last week they were treated there was a report from Jean Benedict Penn, fellow eye treatment, no immunogenicity. So re-exposed to the same therapeutic agent, a virus, which is really just 3 proteins around some DNA. But it's a great place, the subretinal space and the eye in general, because of the immune privilege, but also because of our incredible imaging that most people don't know about, and I'll show you some of that, and because we've got objective and subjective functional measurement. We have good ways of measuring vision in almost everybody, except the low vision patient. Low vision patients are incredibly hard to measure vision in, incredibly difficult. The FDA convened a special task force in an open meeting last summer to get a consensus on how to measure vision in very low vision patients. And the consensus was that there's no consensus. So we can't agree how to measure very low vision and visual improvement. But it's, as we get into better and better seeing eyes, we have very, very good ways of doing it. This is the old picture of the back of the eye. So 10 years ago, 12 years ago, 15 years ago, before we started innovating imaging the back of the eye, this was the classic picture, the edge of the optic nerve. You can see maybe the macula there, and this is about a 30-degree view, right? And this is like standing outside your garage at night with the door open in the dark with a bright flashlight, and you're shining the bright flashlight on the back wall. Well, we figured out how to turn the light on in the garage. That's the same eye. That's a retinal detector. Can we turn that light off, if that's possible? This is the retina, the wallpaper coming off the wall. You can see the cotton wallpaper in the same eye.